Welcome to CryoTalk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Eva Amsen, and experts in the field of cryo-electron microscopy.
Today on CryoTalk, we are joined by Rob Kirchdoerfer, Assistant Professor in the Department of Biochemistry and the Institute for Molecular Virology at the University of Wisconsin-Madison. He talks about working on cutting-edge coronavirus research.
And with lots of international attention on coronaviruses, we have had to pivot repeatedly to find areas that we can contribute to the field where I don't have to worry about, a young student being scooped 6 months into their project.
What cryo-EM might be able to do in the future.
I think cryo-EM is well on its way to really looking at more biological structures in situ.
And how he wanted to be a scientist ever since he was a kid.
I was going to be a marine biologist, and then I decided I was going to do genetics because I watched Jurassic Park, and darn it, let's bring back the dinosaurs. And then I went to college and I fell in love with biochemistry.
All in this episode of CryoTalk. Hi, and welcome to CryoTalk. I'm Eva Amsen, and I'm here today with Robert Kirchdoerfer, our first guest of Season 2. Rob is Assistant Professor in the Department of Biochemistry and the Institute for Molecular Virology at the University of Wisconsin-Madison. His research group has been using cryo-EM to study viral proteins, so we'll hear a bit more about that today. Rob, how are you?
I'm great. How are you this afternoon?
Good. Thanks for joining us.
Thank you so much for having me.
Yeah. So we usually ask our guests to start with a little bit of background. So can you tell me a little bit of your career so far?
So I did my bachelor's degree here at the University of Wisconsin-Madison. I got a major in genetics and biochemistry, and I was doing undergraduate research studying protein folding. And for graduate school, I decided I really wanted a higher resolution view of how the world worked. I really wanted to see how proteins interacted with one another. And so I decided that I was going to do structural biology for graduate school. So I went to the Scripps Research Institute in La Jolla, California, and I did my PhD with Ian Wilson with the goal of looking at an influenza polymerase complex with crystallography. We never did solve a structure of an influenza polymerase with crystallography, but we did learn a lot about other interesting pathogenic systems. And we actually wound up collaborating with some really great electron microscopists at Scripps to do electron microscopy of the influenza polymerase. From there, I actually stayed at Scripps for my postdoc, or for both of my postdocs. I did my first postdoc with Erica Ullman-Sapphire doing crystallography of Ebola virus proteins. And then, we came across a problem in Ebola virus that we couldn't solve with crystallography. And so I started collaborating with Andrew Ward, who worked across the street also at Scripps, to learn cryo-electron microscopy to study these Ebola virus proteins, and then kind of transition from there into studying coronavirus proteins. So I've been at UW now for about 3.5 years, starting up my own lab, and we've got a pretty great group growing up.
Great. So yeah, you mentioned coronavirus, and I think you started in Madison in 2019. Is that right?
Yes. So
where you were looking at coronaviruses, and suddenly they became very irrelevant. Is that what happened?
That is exactly what happened. I have noticed a reoccurring theme in my career that I tend to work on viruses that after I start working on them seem to cause pandemics. So I was working on influenza in 2009 before swine flu hit, and I was working on Ebola virus in 2014 when we had the West African Ebola virus outbreak. So I tend to pick viruses that are very medically relevant and have that impact. I don't ever look for an oncoming pandemic.
Maybe if you change your topic, let everyone know well ahead of time.
I have been asked by family members to let them know if I ever start working on a new virus for that very reason.
So how have the last few years been for you working on coronavirus structures?
It's been really busy. So it's been really great for the field in terms of the things that we learned, the advances that get made. We have a lot more technologies that are being used to address these problems with lots of different approaches. In terms of my lab, this has presented a lot of challenges because my lab is very young and very small. And with lots of international attention on coronaviruses, we have had to pivot repeatedly to find areas that we can contribute to the field where I don't have to worry about, a young student being scooped 6 months into their project. So we have been kind of adapting that way, but it's exciting times, and I think that we're really drawing on a lot of the international research that has come out of research on COVID-19.
Yeah, I guess it has its drawbacks to have everyone else in the world working on the same topic at the same time. Yeah, you mentioned when you started using cryo-EM, and how are you using it at the moment? Is there any specific application that you're focusing on?
Yeah, we really do single particle cryo-electron microscopy. So one of the things that my lab specializes in is preparing really highly purified protein samples, and we reconstitute our complexes in vitro, and then really trying to get at how those complexes interact with one another, right? So how does this protein talk to that one? And cryo-EM has been really great for that, because not only can we get the structural information, but we also get, or rather get around some of these issues of heterogeneity. And so with crystallography, you want that rock solid sample that's going to crystallize. But in cryo-EM, you can look at flexible things, you can look at substoichiometric complexes and slightly unassembled complexes. And so it's really amenable for us looking at some of these more dynamic machines.
And what makes it so useful for viral proteins? I know you mentioned earlier that it's kind of like the resolution and the detail. But...
Yeah. So what I love about any structural biology is really being able to see one protein interact with another. For me, the best part of structural biology is when you get that first map, and you can now see your protein in the map. You can actually see, oh, this amino acid contacts that amino acid. I love looking at RNA in electron density. I think that it's beautiful. So yes, I think that we can use cryo-electron microscopy for a lot of things with viral proteins, but I think it's a much broader technique for really looking at any sort of purified biological sample that we want to look at. And of course, the field is expanding, so maybe we don't always need to purify as well as we used to.
And you were working on Ebola before. Are you planning on going back to that? Like, has it all just been coronavirus these last few years because that's what everyone needed to know about at the moment? Or?
Yeah. So the plans always change. The plan when I started up my lab was to go coronas and stick with coronas to kind of maintain a lab focus. We do look to kind of expand to some other viruses, but I think we'll probably stay in the positive sense RNA viruses. So this is going to be other viruses that have similar genome architectures to coronas. And part of that is just to make sure that we keep some common themes in lab and kind of have a more cohesive scientific environment.
So what are some other viruses that would fall in that category?
Well, so we usually think of coronaviruses as, we talk about the coronavirus, but coronaviruses are actually a very large family of viruses. And so one of the themes in our lab is to really look more broadly across the coronavirus family at different species of coronas to kind of get a sense of what is conserved, what are the general themes that evolution has maintained across this broader family. We're also working with a number of groups. We're part of a consortium. This has been funded by NIH to identify antiviral drugs. And so this is a consortium being run out of UNC Chapel Hill with Ralph Baric to look at a number of positive and negative sense RNA viruses, and in particular, trying to figure out how particular small molecule drugs can impact the activity of viral polymerases. And so that's another way that we've kind of expanded our repertoire of viruses.
Yeah, and I guess anything you learn about one virus, that also tells you a bit more about a related virus as well.
It does. I think that that's a major power of structural biology and biochemistry, is being able to draw parallels and even contrasts. One, for solving problems. I have this trick that worked for this protein. I can probably apply it again for this other protein, but then also for biological themes as well, in terms of mechanism and interaction.
And just going back quickly to cryo-EM, is there anything, where do you see cryo-EM going in the future? What do you hope it will be able to do?
I think cryo-EM is well on its way to really looking at more biological structures in situ. Really looking at more cellular structures at higher resolution with better annotation, with higher more sorting of the available heterogeneity. Right now, I think this is mostly limited to relatively large complexes like ribosomes, but I would love to see that applied to smaller and smaller protein complexes. There's going to need to be some technical advancements for that, but I really think that we're on our way to getting there.
Yeah, that would be interesting to just see everything in situ. Changing tactics a little bit, let's talk about not work. What do you do when you're not in the lab and not looking at protein structures?
I'm a gym rat. I go to the gym. I like to swim is one of my sports. Otherwise, I really just like to spend time outdoors, and that could be walking through the woods or sitting on a boat. Just that kind of quiet outdoor time is really what I look forward to.
Have you had a chance to go outdoors a lot lately? I guess it's winter now. But...
Yeah, it's winter in Wisconsin, and it's really cold. Winter's not my season. I look forward to spring and fall. So I still go on walks pretty regularly, but you're kind of bundled up, and there's not a lot of wildlife to look at this time of year.
Yeah. So I've got a few quick fire questions, so you could just answer them really quickly. The first one: do you enjoy traveling?
I am not a big traveler. I am a bit of a homebody. Traveling kind of stresses me out, but I love being other places. So I guess I have a mixed feelings about traveling.
The process and the destination are not the same. Yeah. Do you have any favorite places where you've been?
I love San Diego, which is where I did my PhD work. I've not been back since I left when I was doing my postdoc because of COVID, and I'd love to go back and hang out.
Yeah.
I think that San Diego has some of the nice microbreweries in the country.
That's a reason to go back.
It's a great reason to go back.
Do you enjoy cooking?
I do. I don't get to cook every night, so most of my cooking projects are kind of weekend cooking projects, and they tend to be a bit bigger, so it's like food prep for the week. But recently I've been making my own pasta, and I also make my own yogurt.
Nice. Yeah, I was going to ask if you had any recommendations, but pasta sounds interesting. What about reading? Do you like to read?
I do like to read. So I obviously read a lot at work, which is all nonfiction. But when I get home, I really like to read fiction, and especially fantasy. And so one book that I read this past year it's called A Wizard's Guide to Defensive Baking, which is a slightly humorous touch of magic kind of story. And it's lighter reading, but I really enjoyed it.
It sounds fun from the title alone. I might read that. Yeah. And what about the big and small screen? Do you like film or TV?
I generally do. I've not been to a lot of movies in the last couple of years. And all of my TV is now switched to streaming services. So, I kind of cruise Netflix to see what's new and what's interesting. Right now I'm working my way through Netflix Wednesday, which is kind of dark and fun, but also kind of creepy. So, yeah.
Yeah, I finished watching that not too long ago. That was fun. And do you listen to music?
I do. I like a lot of different types of music. I'm a big fan of kind of the indie rock, the Modest Mouse, Cage the Elephant sort of things, but then I mix in like dance music from Lady Gaga and Kesha, things like that. And then I'll turn it around and I'll put on some instrumental folk music for a change. So it all kind of depends on what mood I'm in.
So is there music playing in your lab, or does everyone just have their own headphones on?
They all wear headphones because I don't let them play music in lab.
Yeah, I think when back in the day when I was in the lab, we used to have the radio on and kind of discuss what we were listening to while working, but it was a bit distracting sometimes.
Yeah.
And this is a question I love to ask people. If you weren't a scientist, what would you be?
Oh, I've wanted to be a scientist for such a long time that I'm going to struggle to answer that. Um, I think I would need to be in a career that I would be helping people. And so that could be something like a teacher, but it also could be something like a doctor. Just because that's where I get my fulfillment from work. Is that even, as a researcher, I'm making incremental advances that could someday help someone. But I still feel like I need to contribute like that.
Yeah. I feel like teacher and doctor are kind of like science adjacent careers.
They are science adjacent, yes.
So that's fun. So you knew even when you were a kid that you wanted to be something related to science one day?
Yeah, I went through a number of different scientific paths as like a fourth grader. So yeah, I was going to be a marine biologist, and then I decided I was going to do genetics because I watched Jurassic Park, and darn it, let's bring back the dinosaurs. And then I went to college and I fell in love with biochemistry. So it's kind of wandered a bit, but it's always kind of been figuring out how the natural world works and what we can really do with it.
Yeah, I also ended up in biochemistry eventually. I didn't really know that it existed until I started in undergrad, because you don't really, in high school, it's just, this is chemistry, this is biology. Nobody tells you that there's an in-between. It's like, oh, I can do both. Yeah, so, and the other question I really love to hear the answer to is if you have any advice for researchers who are just starting out their career.
I have tons of advice.
Feel free to share.
So the thing I always try to tell students when they're making choices about what lab they're going to join and how they're going to do their PhDs is to find something that they're really passionate about and just be really bold. So there's a lot of apprehension, like, oh, can I make it in this field? Can I get that next big experiment to work? And I think that if you really just have something you're excited about and that you can bring your passion, your resilience to, that as long as you are willing to take that step and actually try, that the rest all kind of falls into place. And so, everybody wants a 5-year plan and a 10-year plan about what they're going to do with their lives. And inevitably, all of those plans change. But I think that as long as you're pursuing something you're excited about and you are willing to take those leaps and really chase those dreams, that even if you don't wind up with the dream you thought you were going to have, you're still going to wind up pretty happy. So
that's good advice. Yeah. I think students are also a little bit scared about, if they take a big risk, what if it doesn't work and I don't get results and I can't write my thesis? But you can graduate with negative results. I think people just don't quite realize that.
You absolutely can. And I mean, if you're doing science, if you're testing hypotheses, then it's quite possible your hypothesis was wrong, but that's data too.
Yeah.
So I mean, I wouldn't discard negative data because it's negative. You showed something, you proved something.
Absolutely. I think that brings us to the end of the episode. So thank you very much, Rob. And thanks everyone else for listening to or watching CryoTalk today.
Thank you for listening to CryoTalk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific. To view all audio and video recordings from this series, please visit bitesizebio.com/CryoTalk.